This diagram that we're looking at here is what we refer to as the cell cycle. And the cell cycle is really the life cycle of the cell. This includes both growth portions, and so all the growing that the cell is going to do, and it also includes the reproductive processes. And by reproduction, when we're talking about cells, that really is cell division. So we do take this cycle and we do divide it into two major portions. Interphase is what we're seeing here in gray. And as you can probably tell, interphase is going to take up the vast majority of the cell cycle. So interphase, it is very long. And I like to think of this as the preparation phase. So what I mean by that is this is preparation for cell division. And so all of the things that have to take place before we can actually split the cell into two is going to take place during this interphase, which you're seeing again in gray here. So we're going to start this whole cycle, if we do think of it as having a start and a stop, although it is a cycle, so it goes round and round. If we were to start right here, we would have one cell, and this one cell that I'm looking at, this would be my parent cell. And through all of interphase, if we're moving in this direction, there is going to be a lot of growth that has to take place, so that by the time we get over here to the end of interphase, we're going to have a larger cell. That larger cell will have done all of the preparatory steps that need to be completed, so that once it enters this pink area, which we're going to call the mitotic phase, we will be ready to actually split. So when we go through those and we come out on the other side, if we were to look at what we end up with over here, we're going to have two small cells. And those two small cells, those would be what we refer to as daughter cells. Okay, so another way to look at the cell cycle is how we go from one cell, we grow that cell, copy all of the information inside of that cell, and then finally, when we get to the mitotic phase, we're going to divide that cell into two. So now what we want to do is we want to talk more about the individual phases of interphase and what would be actually occurring in the cell at that particular moment in time. The first stage of interphase is G1. So if we look over here, G1 stands for gap 1. And if we talk about things that are going to happen in gap 1, you'll see that right here it is labeled as cell growth. So this is definitely a growing phase. We are going to find that the cell is going to copy the organelles during this point in time. We're going to make a lot of different enzymes, proteins, things that we will need later for the next phase. And we will see an increase in the cell size as we go from the beginning of G1 to the end of G1. Since we're going to be doing so much inside the cell during this phase, if we were to actually look at a cell that happens to be in G1 interphase, we would find that that cell is going to have a nucleus there, and the DNA is not going to be something that we would be able to actually see inside the nucleus. So the condition of the DNA itself is going to be that it is not condensed. And the fact that it is not condensed means that it would not be visible. So we wouldn't be able to see any DNA inside the nucleus or any chromosomes inside the nucleus, but that definitely doesn't mean that the chromosomes aren't there. Also, if we talk about the condition of those chromosomes inside of that G1 cell, the chromosomes are going to be made up of one chromatid each. So we would see this. So the chromatid itself really is one piece of DNA with the associated proteins. So we haven't copied the information that is there yet. So we look at it, we see one centromere, we see two arms on that single chromatid, which is making up one chromosome. By the time we get to the end of the G1 phase, we expect that we have certainly prepared everything that we need in order to go into the next phase. And the next phase is going to be DNA synthesis. So other things that we prepared during the G1 phase is that we have to prepare really DNA building materials. And what this would include is this is going to include a lot of different nucleotides, that A, T, C, and G that we will need for DNA synthesis, along with a lot of organizing proteins and also enzymes. If we shift over and we talk about the S phase of interphase, S stands for synthesis. So this is where it's a very important phase. This is where we're actually going to copy, or you could say synthesize, the DNA. One thing that we do definitely have to prepare during interphase is we have to copy all of the genetic information so that we will ultimately have 2 copies that we can divide into those 2 cells that we're going to produce. So when we start off this phase, at the very beginning of S phase, we're going to have DNA that's made up of 1 chromatid each, just like we had in G1. But what's going to happen is that during the phase, we're going to really build another copy right on top of that. So as we build it, when we're done, we will actually see that we have 2 chromatids that are attached together. So that would be what we have when we get to this point in the cell cycle. So when we finish S, we expect to have what we would refer to as replicated chromosomes, or you could say duplicated chromosomes. It would mean the same thing here. So I've got now a bunch of chromosomes that look like X's, if you could see them. But again, we're going to be utilizing the information in the DNA. We're actually looking at it and we're using it to make this second copy. And so again, if you were to look at your nucleus, and I'm just drawing the nucleus here, but it would be inside the cell, you're not going to be able to see the chromosomes. They're going to be very unwound, so that all you would see inside that cell, if I did just draw the cell on here so we can remember that it's there, if we were to look inside that cell, we could really just see a nucleus there. So the nucleus would definitely be intact. But again, all those chromosomes are going to look like X's. The 2 sides of this X, this one and this one, the 2 chromatids, they would be identical to each other. So that's identical in every way, which means they contain exactly the same DNA sequence. And the way that we refer to that is we now call them sister chromatids. So the fact that they are sister chromatids means that they are identical to each other. We really used one of them as a template to make the second copy. That's what's going to take place during S phase. S phase, again, is a very crucial phase because we want to make sure that we have 2 exact copies of the DNA before we actually split it into the 2 cells. The last phase of interphase is G2 or gap 2. In G2, it will be a little bit more growth that takes place, but for the most part, we have prepared the materials that we need to divide. So we've copied the organelles, now we've copied the DNA, and we are going to prepare or make some of the proteins that are going to help in the actual division process itself. But we do tend to think of this a lot of times as a checking phase. And by checking, the cell would be actually checking the DNA to ensure that it is correctly copied. And if it's not correctly copied, then we would definitely want to go and we want to fix that. Because again, the overall goal of this cell cycle is to take a parent cell and to produce from that 2 daughter cells that will be identical to each other, and then they'll also be identical to that parent cell that they came from. Okay, so when we do finish interphase, we expect that our chromosomes are still going to look like this because we've just been double checking them. We haven't really changed what we have there. And we're still going to have a cell that visually looks the same as what we had in the other phases. So we'll be able to see the nucleus, but we won't be able to see any chromosomes inside of it. But if we could see the chromosomes inside of it, they would be chromosomes composed of 2 chromatids, which means that they would look like little X's. So if we review again what happens with the chromosomes as we go through the interphase of the cell cycle, when we begin this whole process in G1, this is what we're going to have. We're going to have chromosomes made up of 1 chromatid. When we talk about these chromatids like this, where there's just one of them, these are what a lot of times are called unreplicated chromosomes. Now during G1, we are doing a lot of other things. So we had other materials that we had to copy and we had to grow the overall cell. Now when we get to S phase, so when we proceed and we go to S, in S is where you're going to take this chromatid and you're going to build the second chromatid on top. So by the time we get to the end of S phase, what we see is that we've got 2 copies of it. So at the end of S, we will have chromosomes made of 2 chromatids. And these will then be what we call replicated or duplicated chromosomes. They have really twice the information there. They've just got a copy of it. So again, each of those sides would be what we would call a sister chromatid. They are identical to each other in every way. And then when we go into G2, in G2, we're going to have really the exact same thing we had at the end of S. We're just going to double check it and make sure that everything was copied correctly. So in G2, they are also replicated chromosomes, which means that we've got chromosomes that are made up of 2 chromatids each.